DNA was isolated using the Quiagen kit from our positive and negative samples of DNA that were taken from the cell pellets that was gone over in this page. So we had our negative and positive UV cell pellets, and those were defrosted, and once those were defrosted and we were able to add solution to them, we added 200 microliters of PBS to each our positive and negative DNA. And then we added 20 microliters of Pronase K from the Quiagen kit to each of our samples. And then we added 200 microliters of buffer AL, the lysis buffer, from the sample. And then we vortexed using one of these, both of our samples, and then centrifuged them at 800 RMP for 1 minute. We put the flow from the centrifuge to in the waste and added 200 microliters of buffer AW1. Once we added the AW1 buffer, we vortexed and incubated for 10 minutes at 56 degrees. Once they were done, we were done incubating, we could then add 200 microliters of 100% ethanol to each of our samples. We then vortexed and centrifuged for another minute, and then we added 500 microliters of the AW1 buffer that we added before to each of our samples. We then centrifuged for another minute, and then we added 500 microliters of AW2 buffer from the kit, and we centrifuged on max speed this time for 3 minutes. After that was done, we discarded the waste and added 100 microliters of the buffer AE lysis buffer. We could then incubate for 1 minute after adding this buffer and centrifuge for another minute at 800 RMPs, and then we could use those samples for the nanodrop for DNA quantification in the next step.